It moves with purpose, hunts with precision, and rebuilds its own body, yet it has no brain. The starfish survives through a nervous system unlike any other in the animal kingdom. Starfish, more accurately known as sea stars, belong to the class Asteroidea within the phylum Echinodermata. More than 1,900 species inhabit oceans from shallow tidal pools to abyssal depths exceeding 6,000 meters. Despite their ecological success, sea stars lack a centralized brain. Instead of a single control center, their nervous system is decentralized. A circular nerve ring surrounds the mouth at the center of the body. From this ring extend radial nerve cords along each arm. These nerve cords coordinate movement, feeding behavior, and sensory input without hierarchical command. This distributed arrangement allows each arm to function with partial autonomy. If one arm encounters prey or an obstacle, local neural circuits initiate responses while still communicating with the rest of the body. There is no cortex, no cerebellum, no cranial structure, yet behavior remains coherent. The absence of a brain does not imply simplicity. Rather, it reflects a fundamentally different organizational strategy, one that prioritizes redundancy and resilience over centralization. Movement in sea stars depends on a hydraulic network unique to echinoderms, the water vascular system. Seawater enters through a sieve-like structure called the madreporite, located on the arboreal surface. From there, fluid flows into a ring canal encircling the mouth and into radial canals that extend into each arm. Branching from these canals are hundreds, sometimes thousands, of tube feet. Each tube foot ends in a suction cup-like structure capable of adhesion. Through muscular contraction and fluid pressure changes, tube feet extend and retract in coordinated waves. A typical sea star may possess 1,000 to 2,000 tube feet, each controlled locally by nerve connections from the radial nerve cord. These feet allow locomotion at speeds generally between 10 and 15 centimeters per minute, though rates vary by species. Beyond movement, the water vascular system facilitates respiration and sensory interaction with the environment. Gas exchange occurs primarily through thin-walled structures called papillae, while tube feet detect chemical and tactile cues. This hydraulic mechanism replaces skeletal leverage with fluid dynamics, enabling remarkable strength. Sea stars can pry open bivalve shells using persistent, evenly distributed force, exploiting even millimeter-wide gaps. Many sea stars are opportunistic predators. Species such as Pisaster acraceus, commonly known as the ochre sea star, prey on mussels and barnacles along rocky coastlines. Using coordinated tube feet, they grip and gradually separate the shells of bivalves. Once a small opening forms, the sea star everts its cardiac stomach through its mouth and inserts it into the prey's shell. Digestive enzymes begin external digestion, breaking down soft tissues before ingestion. The partially digested material is then retracted into the body. This feeding strategy requires precise timing and coordination. Chemoreceptors located on tube feet and skin detect dissolved organic compounds, guiding the sea star toward prey. Despite lacking a centralized brain, neural integration across the nerve ring ensures that multiple arms contribute effectively to the task. Sea stars exhibit remarkable persistence. Studies show they can maintain pulling force for hours, gradually exhausting prey. The decentralized nervous system distributes effort across arms, reducing reliance on any single limb. In certain ecosystems, predatory sea stars act as keystone species. Removal experiments conducted along Pacific coastlines demonstrated that eliminating Pisaster acraceus led to dramatic declines in biodiversity, as mussels monopolized available space. The sea star's feeding behavior thus shapes entire communities. One of the most striking adaptations of sea stars is their regenerative capacity. Many species can regrow lost arms, provided a portion of the central disc remains intact. In some genera, such as Lynchia, an entire individual can regenerate from a single detached arm through a process known as fissiparity. Regeneration involves cellular dedifferentiation, where mature cells revert to a more primitive state before proliferating and redifferentiating into specialized tissues. This process may take months to over a year, depending on environmental conditions and species. The decentralized nervous system enhances survival during injury. Because control is distributed, loss of one arm does not paralyze the organism. Neural pathways reorganize, allowing continued locomotion and feeding during regeneration. This capacity reduces mortality from predation. Fish and crustaceans frequently attack arms, yet many sea stars survive such encounters. Energy allocation shifts toward tissue reconstruction, demonstrating a physiological trade-off between growth and repair. Regeneration without a brain underscores the adaptability of decentralized control systems. Although lacking a brain, sea stars possess sensory structures capable of detecting light, touch, and chemical gradients. At the tip of each arm lies an eyespot composed of photoreceptor cells. These simple eyes do not form detailed images but can distinguish light from dark and detect large structures, aiding orientation toward reefs or away from open water. Recent research indicates that some species can resolve spatial information sufficient to navigate complex habitats. Combined input from multiple eyespots allows the animal to interpret environmental contrast patterns. Chemoreception plays a dominant role in prey detection. Dissolved molecules trigger behavioral responses through receptors distributed along tube feet and body surfaces. The nerve ring integrates these signals, coordinating movement without centralized processing. Sea stars also respond to gravity and water currents. Their radial symmetry, typically 5 arms arranged pentaradially, reflects an evolutionary transition from bilateral larvae to radial adults. This symmetry supports multidirectional interaction with the environment, complementing decentralized neural organization. Ocean warming, acidification, and disease events such as sea star wasting syndrome have caused significant population declines in recent decades. Because sea stars often function as keystone predators, their loss can restructure entire ecosystems. Understanding their unique biology is essential for conservation. The absence of a brain has not limited the sea star's evolutionary success. Instead, it has produced a resilient, distributed system capable of movement, predation, regeneration, and environmental awareness. In the quiet intertidal zone, a creature without centralized thought continues to shape the balance of marine life. Thank you for watching. Subscribe for further explorations into the hidden architectures of life beneath the waves, where even brainless bodies reveal extraordinary design.